Today, I have to say, this is my first time at ESPGHAN. I'm an adult gastroenterologist by training, so this conference always happens at the same time as the British Society of Gastroenterology, so it's a great experience for me to be here. I'm normally used to speaking to pediatric gastroenterologists in a transition clinic, where my job is to deal with the consequences of whatever's happened over the last 16 or 18 years, or however long it might be. So to misquote Einstein, I'm standing on the shoulders of giants, but we'll see how we get on. My conflicts of interest are represented here. I have previous research funding and ongoing research funding from my academic interests, and I'm an employee of Nestle. So my key messages, if you want one slide, this is the one slide to take home and think about, is that I would suggest, and I think there's good evidence, that the gut microbiome plays a critical role in energy harvest from dietary glycans, and we'll unpack a bit what I mean by dietary glycans, and also modulates other diet-related health effects. That the progression that we perhaps can and should introduce into dietary glycans will drive microbiome development, and the two will very much go together, and that the microbiome established in early life will persist into adult life, and in that sense, all good? And in that sense, will very much have an impact on what happens for the rest of that child's life. And we'll hear more about that later, I think, as well. I probably don't need to tell this audience that weaning is a key window, but in the sense of the relevance to the microbiome and why it might be key, we'll get into that a little bit. And I think there's always a question with the microbiome about so what and how do we measure success. And I'll talk a little bit about how we might look at that and how we might look at it again in the future. So we'll talk a little bit about formation in the early life microbiome, and I'm very grateful to the state-of-the-art lecture in summarizing much of that in 5 minutes. I'll take a little bit longer to unpack it. We'll talk about glycans in the microbiome development, weaning and measuring success. So I think this message is pretty well replayed, but it's always the best place to start about how early life exposures determine microbiome development. And I'm sure you're familiar with the impact of cesarean section and breastfeeding. I think increasingly we recognize the effect of the surrounding environment, whether that's pets, whether that's siblings, whether that's soil and all the hand-to-mouth activity that's happening in those early years, and how all of those other microbiome exposures may feed into the gut microbiome. The effect of medicines and the effect of the microbiome on medicines is increasingly well appreciated. And of course, we'll move through to diet. For me, I think the key point about how the microbiota matures and develops is that it's dynamic and sequential. In this period, in the first 1,000 days, we can say, and we'll go through that, you have the opportunity to manipulate a system before it becomes stable and structured. And where it is tomorrow will be very much determined by where it is today. So those steps that we take in the journey of microbiome maturity are going to impact a lifetime. And once we get into year 3, year 4, year 5 of life, that we have less opportunity to impact it critically unless there's a significant microbiome event. This is the question that always comes back and to which I don't think anyone has a perfect answer. How do we define a healthy microbiome? If somebody asks you, what do I want my microbiome to look like? These are some of the thoughts that we've had and are borne out in the discussion. Some of which describe the ecosystem of the microbiome and some of which describe specific features of that microbiome. Diversity is a phrase that's bandied about a lot, but sometimes I think hard to understand. For me, the best explanation of diversity I've heard is if you have a bag of balls and you put in and take a red ball out, what are the chances that you'll take out a red ball the next time? If you've got all red balls, then the chances are 100%. If you've got 100 balls of different colors, then it's 1%. That's a more diverse bag than the first bag. Also thinking about the ecosystem, we talk about resilience, the capacity to recover. We want this microbiome to be on a healthy, natural developmental trajectory, as with many other developmental trajectories. How does it overcome any impact that might knock it off course, whether it's antibiotics, infection, or other things? If we look at specific features, we're starting to unpack the keystone species, the ones we really want to focus on and which might be biomarkers for future health. And there's been some discussion about Faecalibacterium prausnitzii as an example, for which I know the IBD community will be familiar with. And one of the key reasons that we focus on that is because it's a short-chain fatty acid producer, and we'll get into the different sorts of metabolites that may come out, but ultimately I think of the microbiome as a mini factory, and it produces metabolites that then have distal signaling effects, or that might be nutritive or bioactive. Which metabolites should we be thinking about and why? And again, I know this is old ground, but sometimes it's worth unpacking why we care about this and the role of breastfeeding in stimulating microbiome development. We talked about diversity as a measure, and you can see, I think if I use the mouse on this side, that in the formula-fed infants in this study, that there's a rapid increase in diversity in the first month of life compared to the breastfed infants, where that increase in diversity is more gradual in the first 6 months of life. The reason for that, I think, is because of the proliferation of bifidobacteria. So imagine my bag of balls again, except now it's a bag of bacteria. If you have a huge load of bifidobacteria in that bag, the chances of you picking out two, one after the other, are much higher. So by definition, a proliferation of bifidobacteria may decrease the measure of diversity. So when you look at these scores, I think it's really important for me to think about where they come from and how you put them together and how you understand the combination of those scores. But what we're looking for here is perhaps a more gradual increase so that the infant and other systems, whether it's the gut or the immune system, have time to develop with the microbiome. I think a really key point about the bifidobacteria and why we're interested in them is that they're short-chain fatty acid producers. So this Bifid Shunt, and sorry, for those of you that love a biochemical pathway, I've taken away the biochemical pathways because I thought that might be a bit heavy, certainly for me. But these shapes represent biochemical pathways. That is the Bifid Shunt that's common to all bifidobacteria, perhaps expressed in different ways. They'll take a complex carbon source and ultimately turn it into acetate or lactate principally. Now, there is some variation in the bifidobacteria as to what can use different carbon sources, and we'll talk about that next. That may be relevant in the development of the microbiome over time. But the key point is that the bifidos are going to produce acetate and lactate, and we'll talk in a bit about why we care. So what are the complex carbon sources, the glycans, that we might be interested in? My previous research has been very much about the role of fiber in gut function, principally in IBS, and then further in physiology and the effect on and the role of the microbiome in that interaction. So I get very twitchy when people talk about fibers, because to me it encompasses a huge range of different types of molecule, and we shouldn't lump them all together. Cellulose is very different to inulin, is very different to starch in its performance. There's a lot in this conference and more in this talk about human milk oligosaccharides and their particular role. They're slightly more complex than lactose, but not so much more complicated. So here we're looking at oligo short-chain saccharides that are perhaps easier to digest than the long-chain molecules as you move into the simple fibers that we get down to the GOS and the FOS, sorry about that typo there, the inulin, and then over time the more complex fibers. The complexity means that it's harder for the bacteria in the gut to access the breakpoints in those compounds, and to digestion. This is a talk about digestion at a digestive diseases conference. And so it's actually understanding the capacity of the microbiome to digest those fibers and turn them into energy sources or bioactives. And for me, it's helpful to think about a progression in the dietary sources or the dietary glycans that are used. So we start with simple molecules, and over time, as we move up and perhaps into cereals in later life, we have that progression into more complex molecules. How do we digest? We use enzymes. I think everyone here knows that. But this is where I think it helps to dig in a bit to not just what the microbes are, but what they do and how they do it. And for this, I want to establish the principle of the Cas-zyme, the carbohydrate active enzymes. Each bacteria will have a different set of Cas-zymes, and therefore a different capacity to use those dietary glycans, the carbon sources, to turn into short-chain fatty acids. The Cas-zymes that you have will determine which fibers, which glycans you can digest. And so one child's microbiome may be able to digest arabinegalactan, but not arabinate mannan. And there is a whole range of fibers behind that as well that I don't think we need to get into, but I think it does help to establish the principle that more is probably better, and people may sort out into particular profiles with time. But the advantage of a diverse microbiome is that you're more likely to have a range of enzymes and therefore be able to deal with a range of dietary glycans as they arrive and use them for something useful. I would argue that it therefore follows that if you have bacteria that can use a particular energy source, then they are more likely to proliferate and therefore mature. So the types of glycan that you feed the child will steer microbiome maturation through early life. And we talk about alpha diversity, the variation within one individual, how many different bacteria might be present, the variety within the population, and over time, and this is the Teddy study that was briefly mentioned earlier, but to look at early determinants of diabetes in the young and see if there was a role for the microbiome, that you see from birth, everyone very much clusters with a typical, depending on route of birth, but a typical bifidobacteria pattern, and then moves through different fecal community types through the ages, gradually becoming more complex, bringing in other clades that you might have heard of, the butyrate producers, the firmicutes, the Bacteroidetes, etc. But as I said earlier, development is sequential, so how you move through that pattern will be determined about your earlier stages. So for me, it's really about guiding people through the process over the first year of life, which certainly in this study they characterized as more developmental, and then into the second year of life becomes more transitional, and as you move into 30 months plus, you start moving towards what is probably going to be your adult microbiome type for the rest of your life. So a gradual progression that we will steer through diet. And I think it's worth just looking at an example of how we characterize that, how we build up the evidence for understanding what we're doing. And I appreciate that I'm asking for some faith here. One of the challenges, I think, for pediatricians is that it's hard to say what will be the consequences of your actions in 20, 30, 40 years' time. Hopefully all of us will have been retired by then and doing something different. To understand the sort of signals that we might be looking at, and I think it's useful to do randomized controlled trials, of course it is. So this is a randomized controlled trial conducted in Eastern Europe with a non-randomized breastfed reference group, and then for those who chose to use infant formula, they were randomized to control or different test groups, replacing lactose in the formula with different amounts of a 5-HMO blend. You can see that not quite all the infants had stool samples available, but a good number, I think, over 100 in each group. This, I apologize, is the complex microbiome plot, but the principal components analysis is the best way that we have to look for signals in the data to understand discrimination between groups, and then we can unpack those principal components and try and understand the microbiota underlying those changes. It's a complex data plot, but we only need to look at the shapes, which are essentially the mean, the average position of those shapes within the plot. The orange shape is the breastfed group, and in principle, we want to be moving nearer to the breastfed group where we can. This trial is still being analyzed for the later stages, so this is a preliminary analysis from the first 6 months, but you can see that over time, the intervention groups gradually move towards the breastfed group, whereas the control group does not. And I won't get into what's underneath that, but our suspicion is that it's going to be about promotion of bifidobacteria, which would be the main effect of having more HMOs present. But I don't want to speculate too far. So we've talked a lot about the first 6 months, and I should mention that in the Teddy study, by 3 months, 50% of infants were exposed to solid food as well. I think this message is well replayed, but let's just talk about it in terms of glycans. This is the point where we're moving from the breastfed to introducing more complex carbohydrates, some of those other fibers that I showed you in the previous slide. And the main principle that I think I would use to summarize is that diet diversity will accelerate microbiome diversity. The more fibers to which an infant is exposed, the more likely it is that a particular bacterial clade will pick that up, proliferate, and then become part of the sustained microbiome. The other change that we see at this point is that we bring in more butyrate producers. One of the roles of acetate and lactate may be to maintain a barrier that is sympathetic to protein exposure to the immune system. So at this point, we're trying to imprint the immune system, get it to learn about the proteins to which it will be exposed through life, either to mount an infection response or for tolerance. And it may be that an excess of butyrate at this point will tighten that barrier. A good thing in later life, we want a nice tight gut barrier, but not necessarily the key aim here. And it may be that we need a mix of those glycans to maintain the bifidoclades producing acetate and lactate, as well as building up our capacity for butyrate production. And I'll let Jan talk about why that might be relevant. So a period of a mixed diet coming in now. We're not just talking about one element, we're talking about a diet. And so for me, this is about the move from the infantile to the juvenile microbiome. We bring in more fibers. This is the extension of that plot I showed you earlier into year 2 and year 3 of life. There's fewer red bars. This column of bifidobacteria gradually fades as kids get older and other clades come to predominate. Over time, we eventually become with a predominance of butyrate production if we're maturing in the way that we want. And the role of, and the necessity for bifidobacteria fades gradually as we get up the age range. But this comes back again to the question about what should the microbiome look like at any given range? What is optimal maturation? What is the right trajectory? The figures on the left as you look are from Jeff Gordon's group from a few years ago, following a series of Malawian kids to see how their microbiome changed over age, and they tied this very much to malnutrition in those with an immature microbiome, because in a very fiber-rich diet, they may not have the capacity to harvest the energy that they need to acquire from that diet. And if you look at the clades that predominate in predicting maturity there, which is the table in the middle, the table in the middle, sorry, it's a little small, but top of the list, Faecalibacterium prausnitzii, which of course we heard of, and I go back to where I started with the butyrate production, ruminant cocci, Doria, they're all butyrate producers, but still a significant role for the bifidobacteria, the lactobacilli. So we have this mix of short-chain fatty acids. The plot on the right is some work that's, I would say, is in preliminary stages, hopefully will be published soon, from our group, trying to straighten that out. You can see that the plots on the left from the Nature paper are sigmoid, which feels right from a growth chart, but this isn't an absolute measure. We're trying to say that this is the right age. So in an ideal plot, you would have a straight line relationship between microbiome age and chronological age. So still a bit of work to be done before we can actually use that and make it meaningful for clinicians, but I think the principle is sound, and we just need a bit more data and some larger, longer data sets. So my key messages again, and I don't know how I'm doing for time, Anne, but I think I'm about right. And you've seen the slide before, so it shouldn't be that hard. The way I think about this is that the role of dietary glycans is to feed, train, and sustain the microbiome. And I think if that's helpful at all in clinical practice, when people are saying, why do I care about fiber? For me, microbiome is part of the narrative that you can build in some of medicine is science and some of it is an art. And I think the challenge for you is to appreciate the science and then use the art to create a meaningful message for patients. I think we know that there's a critical role for the microbiome in harvesting energy and other effects. I hope I've given you some reason to believe that dietary glycans can drive microbiome development, and that's going to be important for when they get to me. We've talked about weaning and how we might intervene there. Can I tell you exactly what success looks like? Not at the moment, but hopefully we'll come back to you soon and discuss it again. Thanks very much.
Thank you very much. It was a very, very nice and clear presentation. We will go straight ahead to the next presenter, Jan Ravisan, and we will have the question and answers at the end of Jan's presentation.